Okay, well, thank you very much. It's great to be here again in 2022 and appreciate the opportunity to speak again and the support from Alliance for Regenerative Medicine. Immusoft is developing B cells as a modality for sustained therapeutic protein delivery. We've selected the B cell as our cell type of focus for two main reasons. The first is its ability to produce protein. They're extraordinarily prolific. They outpace every other cell type in the body, producing thousands of antibodies per second, producing and releasing thousands of antibodies per second. And so we harness that biofactory capability of the B cells, and we program them to produce therapeutic proteins of choice. The second thing that is beautiful about the B cell is that they will naturally engraft in the bone marrow. And so that obviates the need for some of the preconditioning that's associated with other cell therapy types. This is because this novel modality enables us to obviate that preconditioning regimen. We don't have the immunogenicity associated with some of the viral approaches. We think that will allow us to redose, but nonetheless observe durable expression potentially over many years with each given dose. We started down this path of inquiry by programming plasma cells to produce therapeutic antibodies, and then we began to inquire as to whether we could enable plasma cells or program plasma cells to produce other therapeutic protein types, such as enzymes, signaling proteins, and so forth. And we found that given the right circumstances, indeed, we could. And so that was hugely exciting, and I'm sure you can appreciate that under all those different protein types, there are different therapeutic categories that we can pursue, and as such, this is just a tremendous market potential. Pardon me, for this platform. So we're very excited to be clinical stage. We're the pioneers in the space, and accordingly, we're the first into the clinic with our MPS I program, as we announced just a couple of months ago. And last but not least, we're tremendously excited to be partnered with Takeda to explore certain neurometabolic indications. They're a tremendous group to be working with. I am really just proud and pleased to be working with this management team, and without going into everyone's backgrounds and details, suffice it to say they've got deep expertise going from both small biotechs into big pharma, many successes amongst us under many different umbrellas. When we look at the landscape of protein delivery and the different modalities, there's really kind of three main buckets that I like to look at. The first is the virus delivery. What we address in this sphere is the obviation of that immunogenicity associated with virus delivery. And then when we look at the stem cells, we don't require that myeloablation, that busulfan regimen, that we know is very toxic and best avoided if possible. And so because we don't have that immunogenicity, that preconditioning regimen, we anticipate being able to redose these patients, which would be a huge advancement in cell and gene therapy. This third category is enzyme replacement, which, while not an advanced therapy, is a modality for therapeutic protein delivery that we think actually is really exciting, and there are some great treatments here, but there's still room for improvement that we think our approach will address. And so one of those is that these proteins have very short half-lives, and so the patients need to go in frequently, generally weekly, for their infusions, which impacts their quality of life. And so to be able to reduce that would be a substantial improvement in their QOL. And then moreover, there's a sawtooth effect associated with a drop in the glycosaminoglycans, for example, with MPS I, that starts to raise over the course of the week and then drop again with each subsequent transfusion, or infusion, pardon me. So we think that by 24/7 delivery of the enzyme, that will ameliorate that sawtooth effect and ultimately have better outcomes for these patients. And so our manufacturing process starts with the apheresis of the patient. From that, we isolate out the B cells. We program those B cells in vitro using the Sleeping Beauty transposon system. From there, we expand the number of B cells, differentiate them towards plasma cells, and that's our drug product that we ultimately then inject into the patient. And once that plasma cell engrafts in the bone marrow, we know that it has a 17-year half-life based on the published literature. And so again, we anticipate having many years of biological activity and therapeutic benefit from a given dose. We started out in this endeavor by programming B cells to produce anti-HIV antibodies. And once we got that down, we started moving into other protein types, namely enzymes, signaling proteins, et cetera. And if we look at the different implications, we see that we can move into lysosomal storage diseases, oncology, autoimmune, infectious disease, and so forth. The upshot of that is there's really more than we can undertake as a given small entity, but we're interested in partnering discussions. I'll plug that early, and so. So one of the the vertical I like to kind of drill in on a little bit further is enzyme replacement approaches, and so this isn't a pipeline slide, but what we see here is a number of proteins that have already been approved by the FDA and so have demonstrated safety, efficacy. And that element of the technology risk has been, by and large, removed from the equation. These all, nonetheless, still have unmet need. Each of these programs is in the hundreds of millions range and collectively north of 5 billion. So really just one interesting and exciting opportunity. So that takes us to our pipeline. We're starting with MPS1 for delivering iduronidase to these patients. From there, we move to MPS2, where we're very excited to be partnered with CIRM. CIRM has brought great resources to the program in terms of not just capital, but intellectual capital and expertise. The next program is our follistatin program for muscle wasting. And follistatin is a protein that's released from the liver when we work out. Put plainly, it's a muscle-building protein. It's naturally occurring. What's nice about that is it's also genotype independent. And so we have a potential for application across numerous different therapeutic categories with this program. And so from there, we move into additional ERT approaches, and then cardiovascular, autoimmune, and CNS. And very pleased to be partnered with Takeda in the CNS work. So a little bit about MPS I. It's characterized by the loss or deficiency of the iduronidase enzyme. Iduronidase is involved in the breakdown of glycosaminoglycans. And when those glycosaminoglycans are not broken down properly, they start to build up in the lysosome. They're harmful at the cellular level, and then by extension, the tissue, organs, and organ systems. And so without appropriate treatment, these patients will generally not live past their first decade of life. There are treatments in the way of enzyme replacement and stem cell transplant, depending on the severity of that patient. But what we address is the overall exposure to the enzyme. And by that, it's a function of the protein level times the duration of exposure. And so in the case of the enzyme replacement approaches, we know they have a couple of hours half-life. In the case of stem cell transplant, it's 24/7, but at very low levels. And so some of those tissues are not well penetrated. And so we anticipate being able to deliver higher levels in HSCT, but around the clock, 24/7. And so looking at a snapshot of data, what we have on the left is human IgG. This is in a mouse model. Our drug product is the human B cell, and so we use human IgG as a proxy for engraftment. And so while we see a drop over the course of the 6 months, what we observe in the tissues to the right, let me see if I can do this. Yeah, we've got a 3-month in the red and 6-month data in the blue, and then the black is a non-dystrophic animal. So in the liver, kidney, spleen, lung, we see supernormal levels of iduronidase in the in those tissues at 6 months. We also see some expression in the heart and brain, which is intriguing. And then if we look at the negative correlate, which is the GAG levels, what we're seeing here is complete reductions essentially to normal in the liver at both 3 and 6 months, and then really substantial reductions as well in the kidney, heart, and lung. And the heart is a particularly difficult tissue to penetrate, so we're really excited by that data. We see something going on in the brain which is intriguing, and we've got concerted efforts to increase the expression in the brain. So in the way of our timeline, we're looking forward to dosing the first patient early 2023. We've got a star there to call out the first sign of biological activity. We define that as an increase in iduronidase in the plasma, and or reductions of glycosaminoglycans in the urine. Either of those would be a huge win, not just for this platform, for this program, but for the platform as a whole. And then following that is our MPS II study, which we're looking at entering the clinic either later this year, or later 2023 or early 24. The IDS program for MPS II is coming along very nicely. This is results from a dose-ranging study. What we see across all doses is an increase in the enzyme level in the plasma. And then if we look to the right of that is the reduction of glycosaminoglycan levels, and we do see a nice reduction of glycosaminoglycans as well. The pink being our high dose is correcting to almost normal. The follistatin approach, to keep it simple, the orange dots are treated animals. We're looking at weight gain as a proxy for muscle mass, and we see that the treated animals all have an increase in weight, and we can measure that follistatin level in the plasma, which is important. And then the right graph is the strength in the tibialis anterior, the contractile force, and we see directionally there's an improvement in contractile force. And so taken together, that's exceedingly strong support for our follistatin program. And then when we contemplate those three programs together, what we see is that we can reproducibly manufacture these cells. Those cells produce those proteins in vivo. We can measure that protein in the plasma, and we can measure a therapeutic effect. So very strong support for the platform as a whole. Manufacturing, I'll keep it short and just say we've been approved to go into the clinic with our program. Been a lot of resources, blood, sweat, and tears put into our manufacturing program, but we're very proud with the reproducibility of that. And along the way, we've developed substantial know-how, intellectual property, and filed some patents on elements of our manufacturing. And so speaking of patents, we've got an outstanding patent position with 60 approved and or pending patents. It started with work out of David Baltimore's lab at Caltech. We've built on that internally. We've also in-licensed some IP around the Sleeping Beauty transposon system. We've got freedom to operate as determined by a couple of independent investors, and we've got IP protection out through 2035. And so to sum it up, this is an innovative platform using B cells as a modality for sustained therapeutic protein delivery. Huge market potential across many different therapeutic categories. We're really excited by our partnership with Takeda in exploring the CNS, and interested in discussions should somebody be interested in learning more. If so, please contact me at my email address here. Thanks very much.